It's A-level. We are looking at 16.7 and chapter 16 as a whole, really. We're looking at the structure of the nephron. We know that the nephron is the basic unit of the kidney and it exists over 2 layers of the kidney. We know that the kidney has its structure like this, that we have the outer cortex, that's this region here, and I always say outer cortex because then you don't get it mixed up with the inner medulla, which is here. And the nephrons kind of form collectively these things called pyramids, and ultimately they enter into these things here that is called the pelvis. The nephrons look like this. They are situated with the renal capsule, which is the glomerulus, and the Bowman's capsule in the outer cortex, with the proximal convoluted tubule, which is next to it. The loop of Henle extends right into the inner medulla, and then it goes into the distal convoluted tube and the collecting duct. And the one thing to remember is the length of the loop of Henle is proportional to the concentration of urine produced. So therefore, desert-dwelling animals, like a desert rat, for instance, will have a really, really long loop of Henle because it wants to reabsorb a lot of water. So let's go through it from the top. Here I'll go quite quickly because you can always stop it and slow down the video and have another look. So here we have the blood capillary, and it goes into this knot. It's called a glomerulus, and what you'll find is that the capillary going in is thicker than the one coming out. This is called the afferent capillary, and this is the efferent. And I always remember which way around it is because E for exit. Now the fact that it's wider going into the glomerulus and narrower on the way out creates this really high pressure. It is hydrostatic pressure because it's liquid within. So we have really high hydrostatic pressure. And what that pressure does is it forces the components of the blood out on the basis of their size. So anything that's small enough will pass out through these membranes into this structure called the Bowman's capsule. So what are we talking about? We're talking about ions, we're talking about sodium ions, chloride ions, glucose, urea, water. But the red blood cells and the large plasma proteins are left behind inside this capillary, which then exits here. Now, although we've got this really high hydrostatic pressure causing ultrafiltration, we also have some opposing forces acting in the other way. So, for instance, the endothelial membrane of the capillary. Remember, the capillary is formed by lots of flattened cells that are very, very thin. And as the liquid part of the blood, the plasma, if you like, is being forced across here, then this is going to offer some resistance back. Additional to that, these cells don't just join here by their sticky ends, they also have like a connective tissue holding them together. So that connective tissue, that basement membrane, is also going to offer some resistance back. Additional to that, we then have the cells that line this Bowman's capsule. Those cells are specialized cells, they're called podocytes. And the reason why is because they have pod-shaped structures like these. When they join together, they create gaps, which then aids the process of moving the products from ultrafiltration across there and into this Bowman's capsule. However, they also offer some resistance back against that. You're also trying to squeeze fluid into an area where there's also fluid. So you've got hydrostatic pressure pushing back also, so there's another thing pushing in the opposite direction. And additional to that, I want you to consider this. So when the blood comes through this capillary and the water, the ions, the glucose, the urea are lost into this Bowman's capsule by ultrafiltration, what is left behind is a very low level of water and a very high level of those blood components that couldn't leave. In other words, the plasma proteins and red blood cells. And they exert a water potential, okay? So inside there, we've got a really negative water potential. So in the latter half of this glomerulus here, we've lost the water, we've increased the water potential, and that creates a pull back. So not only have we got hydrostatic pressure acting in one direction, we said we've got the capillary epithelial cells pushing back, offering a resistance, the connective tissue of those epithelial cells pushing back, the podocytes themselves pushing back, the fact that there's liquid inside the Bowman's capsule pushing back, and additional to that, we've got a very low water potential in here, which is exerting a pull for the water to move back into that blood capillary. Of course, the whole thing is so powerful and it's so fast, it doesn't really get much of a chance to do that before the filtrate is moved on through this proximal convoluted tubule. Now, the proximal convoluted tubule is exactly what it says on the tin. Proximal next to convoluted, twisted, tubule, tube. So let's just have a little look at one of the cells that would line that tubule. It kind of looks like this. It is adapted to its function because it's got a lot of microvilli. When they come down, the basement part of the cell has these invaginations. And of course, the blood capillary would be flowing by like this. So this is where our filtrate is, and our cell, and our blood capillary. Because don't forget, this blood capillary now continues, it doesn't just disappear, and it wraps all around this nephron. And you have to remember that for the whole process to make sense. So what happens? Well, this is where we absorb most of our glucose back into the blood. And of course, following the glucose is the water by osmosis. Remember, in biology, we always say where things move from, where they move to, and the process by which they get there. So here, it's very clever. What we do is these cells are packed with mitochondria, and they fuel active transport. Active transport acts on this basement membrane. And what it does is it actively transports sodium ions into the blood. Now, by moving those sodium ions, what we're actually doing is creating a low concentration of sodium inside the cell. And so sodium wants to move in from the filtrate into the cell by diffusion. The only way it can do that is by using a carrier transport molecule that is on this surface here. And as you know, a carrier co-transport molecule also transports something across with it. Now, in this case, with the sodium is brought glucose, with the sodium is brought chloride ions, and with the sodium is brought amino acids. So all of those things are reabsorbed into the blood. So is glucose reabsorbed actively? It's indirectly absorbed actively. It wouldn't be absorbed unless the sodium was being actively absorbed into the blood. Okay, moving on quickly. So, we now look at the loop of Henle. I want you to remember three things. This one on the way down is permeable to water. I want you to remember that this one on the way up is impermeable to water. And that the bottom bit here that I've signified in red has the lowest water potential. Okay, so that's where it's at its lowest. Now the next thing I want you to remember is that when we describe what goes on in this loop of Henle, what we're always going to do is start our story here, because the things that happen on this side are a result of what happens here. So let's have a little look. We start by actively moving sodium ions out from the filtrate into the interstitial space. Okay, so by moving it over, we increase the concentration of sodium here. So before we move forward, let's just have a little think about what will happen here. This signifies really high solutes and really low water. So as we move up here, if we're moving sodium ions out, this is going to start decreasing. When we come over to this side, what we're going to do is we've got a high concentration of salts here, and so what we're going to do is water is going to follow by osmosis. And of course, the key thing you've got to remember is that all of this is got blood capillaries on the outside. So the water moves into the interstitial space and then moves by osmosis into the blood capillary and off. So what's happening here? Well, the filtrate coming from first being moved out of the glomerulus is going to be really rather low in solute concentration, and it's going to have a high water potential. Let's call it 300. As we move down, if we're moving water out, we are going to increase that until we get to the lowest there. As it moves up, we move the solutes out, so therefore it becomes a higher water potential. Then we get on to the business of the distal convoluted tube. Distal, distance, convoluted, twisted, tubule, tube. We are looking at the distal convoluted tubule, this region here. And this is where it gets really cool. This is the area that is under the influence of antidiuretic hormone. Now, this hormone is produced in the hypothalamus of the brain, and it's stored in the pituitary gland. So what happens is, in the hypothalamus, if I just imagine this is the hypothalamus, and then this is the pituitary gland here, in the hypothalamus you've got these cells. And when you start to get thirsty, in other words, when there's less water in your blood, these cells start to shrink and shrivel a little bit. Now, that then sends a signal down to the pituitary gland, and the vesicles that store antidiuretic hormone are then fused and release the antidiuretic hormone into the blood, travels all around the body, but it acts on the cells that line this distal convoluted tubule. The effect that it has is, so let's imagine we're looking at one of those cells that lines the distal convoluted tubule. This would be the filtrate in this region here, and this is the blood that's bringing our ADH, our antidiuretic hormone, from the pituitary. Remember in biology where things move from, where they move to, and the process by which they get there. So here is my antidiuretic hormone, these blobs of yellow if you like, and here is the cell in the distal convoluted tubule. So these here are receptors, and what's going to happen is this antidiuretic hormone is going to bind with the receptor on the cell of the distal convoluted tubule. Now inside that cell you've got an inactive enzyme, it's called phosphorylase, and when that binding occurs, turns the inactive phosphorylase to active phosphorylase. That then moves in the cell and causes these vesicles to move and fuse with the cell membrane. So what do these vesicles do? Well, they contain special carrier proteins inside their own membrane. And these carrier proteins are proteins for water. They are water porins, aquaporins. So when these vesicles move and fuse, they put more channels in the membrane to absorb more water. And the way to remember it is that ADH adds aquaporins. ADH adds aquaporins. If you're adding aquaporins, you're adding water channels. That increases the permeability of the collecting duct to water, which means that water is able to move in back into the blood and help to restore the water potential. And we say restore the water potential and not of the blood, and not raise the water potential of the blood, because remember, where is that blood coming from? It's coming from here. It's had its water potential made very, very negative, and then as it's passing back over here, reabsorbing the water, which helps to restore it to the original where it started. So in an exam situation, we can't say it helps to raise the water potential of the blood, because that would be wrong. We're talking about the blood as a whole. All we're saying is that it helps to restore the water potential of the blood in that capillary that's travelling through the kidney. So let's go back to this diagram here. We've now reabsorbed a lot of our water through the distal convoluted tubule under the influence of antidiuretic hormone. So now as the filtrate moves through, we want to continue to absorb the last bits of water. How do we do that? Well, we've already said that we've got filtrate moving in that direction, and coming down here we've got filtrate moving in that direction. It is counter current. So although the filtrate moving through here now has a really high water potential, what's going to happen is water is going to still move out, because on this side it's a little bit more negative. So water's going to move out from the filtrate into the interstitial space, and then of course off into the blood capillary, which isn't shown on here. When it then increases again, what we're going to get is a concentration gradient continues to remain in this counter current system. We then make the solutes more concentrated again, water continues to move out, and again, water continues to move out. So the diffusion gradient is maintained along the length of the collecting duct. Why? Because we have a salt, an ion gradient that exists in this interstitial space, which is our ion gradient, and the countercurrent mechanism ensures that the diffusion gradient is maintained along the whole length of that collecting duct. And in fact, that is why at the beginning of the video we mentioned that the length of the loop of Henle is proportional to the concentration of urea, the concentration of urine that is made, because the longer that is, then the longer that is, and the more time there is for water to be reabsorbed. Of course, the whole system is a compromise, and with the reabsorption of the water, we get urea. However, the number of times that the blood is filtered through the kidney, the reabsorption of water, we manage to concentrate the urine, so ultimately we do get rid and we do excrete urine from our system. So let's just recap on some of the main points of this whole loop of Henle kidney thing. What we said was we've got hydrostatic pressure, not just pressure, hydrostatic pressure, forcing components of the blood out based on the idea of their size. And we said that against that hydrostatic pressure, there were some elements of resistance. What were they? They were the epithelial layer of the capillary cell, its basement membrane, the podocytes, the fact that there was fluid in there that already had a hydrostatic pressure of its own, but also the decreased water potential, which created a pull, an osmotic pull, pulling the water back. We said that we've got selective reabsorption in the proximal convoluted tube, indirectly reabsorbing glucose, chloride ions, and amino acids by active transport indirectly, because of course with the movement of sodium out, we have the carrier proteins, which brings in with co-transport, by facilitated diffusion, the other products as well. We said with the loop of Henle, you're always going to start your explanation here. Why? Because what goes on here is decided by what goes on here. The pumping out of the sodium ions causes water to follow, and the big idea that all of this is covered with the capillary. Which capillary? The one that's come from this glomerular loop here. And so it's already very, very low in water, and water's going to move readily back in by osmosis. We talked about this cool idea here of the vesicles and the aquaporins, and the ADH, antidiuretic hormone, adds aquaporins. So if you're adding aquaporins, you're wanting to reabsorb the water. So how is that all controlled? Well, when you're low of water in your system and you want to reabsorb it, your cells in your hypothalamus shrink. They then send a message to the pituitary where the ADH is stored, not made, and that is released into the blood, which causes those aquaporins to be in there. And then, of course, we talked about how this countercurrent exchange ensures that there's a diffusion gradient along the whole length of the collecting duct there, and that is how we absorb, reabsorb rather, as much water as possible.